Hello dear students, welcome back to biology class. Chapter 6 Molecular Basis of Inheritance. In the previous video, we discussed about structure of polynucleotide chain. In this video, we are going to discuss about structure of DNA. Discovery of double helix structure of DNA. In 1953, James Watson and Francis Crick proposed a double helix structure of DNA. Before their discovery, there were another two scientists, Maurice Wilkin and Rosalind Franklin, studied the X-ray diffraction structure of an DNA. Based on their studies, Watson and Crick discovered the double helix structure of DNA. Here, X-ray diffraction means molecular study of any cellular material. Next, we discuss about double helix structure of DNA. DNA is made up of two polynucleotide chains. Both the polynucleotide chains running anti-parallel to each other. Anti-parallel means one chain run from 5' to 3' orientation and other chain run in opposite direction and has 3' to 5' orientation. Both the chains are helically coiled with each other and they just looks like a coiled ladder, and here the coiling is right-handed fashion. DNA takes one complete turn, that is from here to here. One complete turn measures about 3.4 nanometer or 34 angstrom, and each turn composed of 10 nitrogen base pairs. And then what is the distance between two nitrogen base pairs? The DNA takes one complete turn at 3.4 nanometer or 34 angstrom, and the number of nitrogen base pair is 10. Here we have to divide the distance with that of number of nitrogen bases. 3.4 nanometer or 34 angstrom divided by 10, that gives 0.34 nanometer or 3.4 angstrom. Hence, the distance between two nitrogen bases is 0.34 nanometer or 3.4 angstrom. Next, the diameter of the DNA, that is from here to here, the diameter of the DNA is 20 angstrom or 2 nanometer. There are two grooves in the helix. The width of one groove is large and this groove is called major groove, measures about 2.2 nanometer. The width of other groove is less, called minor groove, measures about 1.2 nanometer. The backbone of polynucleotide chain is formed of pentose sugar and phosphate group. Inside it consists of nitrogen bases right angle to them. Each polynucleotide chain is formed of nucleotide. Each nucleotide consists of three components: pentose sugar, phosphate group and nitrogen base. Likewise, many nucleotides join together to form an polynucleotide chain. As you know that DNA is made up of two polynucleotide chains. The other chain is running in opposite direction. Hence, there are two polynucleotide chains in a DNA. The next question arises, how these two polynucleotide chains are get binds. There is a specific pairing between the nitrogen bases of two chains. Always a specific purine pairs with specific pyrimidine. Adenine of one strand pairs with thymine of other strand, or thymine pairs with adenine by means of two hydrogen bonds. Guanine pairs with cytosine, or cytosine pairs with guanine by means of three hydrogen bonds. Hence, the two chains are complementary in nature. Here, the fifth carbon atom having the free phosphate group in opposite direction. Likewise, the third carbon atom having the free hydroxyl group in the opposite direction. Hence, one chain has 3' to 5' end, the other chain has 3' to 5' end. Hence, the polynucleotide chains of DNA are called antiparallel, and also they are complementary in nature. Within the DNA, there are three types of linkages are present. The phosphodiester linkage, which links two adjacent nucleotides. Likewise, there is another one bonding, the second bond, N-glycosidic linkage present between the pentose sugar and the nitrogen base. The third bond is the hydrogen bond present between the two polynucleotide chains. In this slide, we see both helical as well as chemical structure of the DNA. Already all the structural characteristics of DNA double helix we discussed in previous slides. Here I am going to tell you some peculiar characteristics of DNA structure. Those points are helpful for your competitive examinations. As you know that the backbone of DNA is formed of pentose sugar and phosphate, and inside it consists of nitrogen bases. Hence, the DNA looks like a ladder. Here, the purines pairs with the pyrimidines. Adenine is a purine, thymine is a pyrimidine. Then the question arises, why adenine not pairs with the other purine, that is with the guanine? Why adenine not pairs with the guanine, or why thymine not pairs with the cytosine? This is because if, as you know that purines are double ringed, the purines are double ringed and pyrimidines are single ringed structure. If purines bind with each other, then in this region the diameter of the DNA is become more, and if pyrimidines are single ringed structure, in that region the diameter of the DNA is become less. To maintain the constant diameter of the DNA, the base pairing occurs between the purines and the pyrimidines. Adenine pairs with thymine, vice versa. Guanine pairs with cytosine, vice versa. The next point is that why the DNA are running in antiparallel. One chain run from 5' to 3' and other chain run from 3' to 5'. Why in antiparallel? Why both the chains not run in 5' to 3' end? This is because of the hydrogen bonds which are present between the nitrogen bases. the hydrogen bonds are get formed between the particular nitrogen atoms of nitrogen bases. Because of the hydrogen bonds, the DNA polynucleotide chains are anti-parallel in nature. Next, we discuss about central dogma of flow of genetic information. The DNA copied to DNA by a process called replication. The one strand of DNA get copied to RNA by a process called transcription. The codes which are present on an mRNA involved in the synthesis of proteins by a process called translation. In this way, the genetic information which is situated on the DNA transferred to RNA and RNA involved in the synthesis of proteins, and this one we called it as a flow of genetic information from DNA to RNA and from RNA to proteins. One good example is in the fifth topic you discussed about the tall character, dwarf character of an pea plant. The genes which are responsible for tallness of an pea plant, they are situated on the DNA. Those genes are copied to form an RNA, and that RNA results into synthesis of proteins which are responsible for the tallness of an pea plant. Hence, by the production of this protein, the tall character is expressed in the pea plant. This one we called it as a flow of genetic information. Irwin Chargaff rule. There was a scientist, Irwin Chargaff, who first time studied about the amount of purines and pyrimidines of a double helix DNA. According to the Irwin Chargaff rule, for a double stranded DNA, the ratio between adenine and thymine and guanine and cytosine are constant and equals 1, or the amount of adenine guanine, that is purines, and ratio with that of amount of pyrimidines, that is thymine and cytosine, equals 1, where the amount of adenine is always equals to amount of thymine. The amount of cytosine is always equals to the amount of guanine. Here in this diagram we can find the amount of adenine, thymine, cytosine and guanine. The number of adenines 1, 2, 3, 4, 5. Adenine are 5 in number. Like that, thymine 1, 2, 3, 4, 5. Number of thymine is also same, 5. Like that, the number of cytosine and guanine 1, 2, 3, 4, 5, 6. The number of cytosine is 6. Guanine 1, 2, 3, 4, 5, 6. The number of cytosine and guanine is also same. Here the amount of adenine and thymine is equal and the amount of cytosine and guanine is equal, and their ratio of adenine and thymine 5 by 5 as well as the ratio of cytosine and guanine 6 by 6 is equals to 1. This is Irwin Chargaff rule. Let's discuss some problems on structure of DNA. Here the base sequence of one strand of DNA, that is 5' to 3' end of the DNA is given. Next we have to find the base sequence of the other strand, and this question can be solved based on base complementary nature of the DNA. Always adenine pairs with thymine or thymine pairs with adenine, guanine pairs with cytosine or cytosine pairs with the guanine. Adenine means here the thymine. Thymine pairs with adenine, guanine pairs with cytosine, cytosine pairs with guanine, thymine pairs with adenine, adenine pairs with thymine, cytosine pairs with guanine, thymine pairs with adenine, guanine pairs with cytosine, adenine pairs with thymine, cytosine pairs with guanine, thymine pairs with adenine. The next question, in a double-stranded DNA of an organism, 17% of the bases were shown to be cytosine. What will be the percentage of the other 3 bases, that is adenine, guanine and thymine? This problem can be solved based on Chargaff rule. According to the Chargaff rule, amount of adenine is equal to thymine and amount of cytosine is equal to guanine. Here they have given the percentage of cytosine amount, that is 17%. That means cytosine amount is equal to guanine, hence guanine amount is also 17%. Cytosine plus guanine is equal to 37%. Then what will be the percentage of adenine and thymine? Percentage means 100. We have to subtract 34 from the 100. We get the 66%. Amount of adenine and thymine is equal. That means 66 divided by 2. We get the amount of adenine and thymine as 33%. Answer is amount of guanine is 17%, adenine is 33% and thymine is 33%. Thank you friends for watching the video.